Guys, so now we're going to talk about eukaryotic cells. So eukaryotic cells have an enclosed membrane. So these guys have compartments. So the best thing I can tell you about the difference between prokaryotes and eukaryotes is, okay, let's compare a single roomed classroom or school to Lone Star. Imagine how hard, say you went to Lone Star and you wanted to actually be a student at Lone Star. And you walked in, or say Lone Star was one room, like it was when it initially started, and compare it to now, right? The one room, you could go in there, and if you wanted to be a student, it would be easy to find the person to register you. And it would be easy to find the advisor to get you in your class. And it would be easy then to pay for your class. And it would be easy to go to class because it would be a very small room, and those people would either be the same person or they would be different people, but it would be easy to find them within that small room. Now let's scale that up to Lone Star. And imagine if Lone Star had the separate floors, but within those floors had no walls. And so you go in our normal situation, you can go to the admissions office, and it's very easy. It has its own room. It has its own employees in that room, so you know where to go. Well, imagine if you came through the front door, and then all of a sudden, you didn't have that room. And that person from admission could be anywhere from level 1 to level 8. And you would just have to walk around and they wouldn't really have anything on them so you could really tell who they are. So you'd end up having to ask everybody, are you admissions? Are you admissions? And that may take you one day. It could take you a couple weeks to just actually be part of the college because the school now is too big to essentially not have individual rooms and or compartments. So that's essentially how eukaryotic cells are. They're too big, so essentially we have organelles to help us with that. So within eukaryotic cells, you have a couple common cells in here. You have animal cells. And so these guys, like us, they're going to be have only cell membrane, so you can see these are cheek cells. You're going to be able to look at them later. You can see in here that on the outside they have the cell membrane. Here you can actually see a nucleus. Those little dots, the dots are mitochondria, right? And so they actually have organelles. That whole fluid, right, is the cytosol itself. If you remember, the cytoplasm is when you have the cytosol plus all the components in it, right? You're going to have centrioles in animal cells. You do not have centrioles in plant cells. And then they also have cilia and flagella. And here are both a cheek cell and a nerve cell. And this is what it looks like, right? And so on here, these are all the different, so you should be able to label and do all the different compartments here. This whole thing right here is a nucleus, right? That is the control center that contains the DNA. Within that, you have these guys right here. Let me get a different color here. Let me get a highlighter. Oops. Let's not do a highlighter. So you're going to have that right there. Those little round circles are nuclear pores. That's for things to get in and out inside the nucleus. This right here is the nucleolus. It actually helps us make ribosomal RNA. These little squiggly lines all in here, that's the DNA, but it's in the form of chromatin. So if you know chromosome, that should help you with the beginning of it, chromatin. And then the 10, I think the N as in normal. You'll figure that how the DNA can be held in chromatin or chromatid. And then this purple here is the nuclear envelope. And that's just the cell layer that essentially is going to divide the nucleus from the rest of the cell. Now, the rough ER is right here in this whole area right here, around here. And essentially that has little dots on it. And the little dots that you see here and all throughout, those are ribosomes. And ribosomes allow you to make proteins. And if you look out here, there are ribosomes there as well. Those are free floating. And those ribosomes, depending on if they're on the ER or if they're free floating, the proteins go different places. Now, right next to it, that ends up looking smooth, is the smooth endoplasmic reticulum. That helps to essentially make lipids. You can see here is the plasma membrane on the outside, and right underneath that, that help with structure are the microfilaments. Over here is the Golgi complex. Essentially what the Golgi complex is going to do, it's going to modify and then move proteins around. They essentially are going to come from the rough ER that helps to synthesize and modify proteins. And we're going to talk more about how they work together in the endomembrane system. These little guys here, along with the blue ones, are vesicles. They're like little trucks that carry things around. Centrosomes, or centrioles, are these little churro-like things here. And essentially, they're also known as microtubules, or they end up irradiating what's called microtubules, are those little lines that come out, or spindle fibers, and they help to divide the chromosomes during cell division. And so that's the microtubules there. The little lysosomes here help, they contain an enzyme that helps with digestion of many of the complex molecules and things that no longer work anymore. The little bean, the mitochondria up here, or chondrion, essentially allow for ATP production. And there are things that are a little bit larger that can be called peroxisomes. And those help to basically get rid of hydrogen peroxide and oxygens that are just kind of rogue. We'll talk more about those. So those kind of go over all the organelles. In plant cells, these guys are really rigid. They have things called plastids that help with energy. They have a large central vacuole. Now, to let you know, the large central vacuole in plastids, animal cells do not have. Okay? And these guys don't have centrioles. And so you should be able to identify this plant model. And I'm not going to go over all the different parts of it because what they do in the plant is the same as they do in the animal. I will point out this is a central vacuole here. That helps to basically store water and other enzyme for digestion. And it helps to give support because we're going to find out later that this thing, as it's full, pushes out against the cell wall and keeps the cell membrane, attached to this guy right here, which is called the cell wall. Now, we do have plastids. The one they're showing here are chloroplasts. And essentially, that is the place of where photosynthesis occurs. And that's where we take light and make it into sugars. They also have these things called plasmodesmata, and those are small channels that allow things to get through the cell wall. And just to let you know, this central vacuole here is also known as a tonoplast. So the cell wall is very rigid. If you remember, it is essentially made up of cellulose. So cellulose is that fiber that essentially is that carbohydrate that is important in structure. And so this is the dietary fiber. And cell walls not only help protect, it help them maintain shape, and it keeps the cell from absorbing too much water or bursting if it does. Right? It won't happen. So the membrane structure, the plasma membrane is important because it helps to separate the cells, from the living cell, from non-living surroundings. And so when we go over, you've learned about lipids, specifically phospholipids. And so essentially the lipids arrange in what's called this bilayer. Right, this bilayer. And so here on the outside, you're going to have their polar heads. And within the inside are the non-polar tails. And so essentially that's going to help separate the outside from the inside, because only few things can get through this guy. Things like gases are very, very small molecules. Water will leak through it a little bit, and you can get things that basically what they keep out are things that are really big, and things that are charged can't get through here, like ions. So it helps to divide cells into compartments. It helps because essentially vesicles can be made from cell membranes, and that helps transport material around. Sometimes proteins are located in those membranes, and they help attach to some other organelle or the cell, the plasma membrane on the outside of the cell. They're important in storing energy and conversion of energy, because when we talk about the mitochondria, the enzymes for energy are located in the membrane. And then they're important in what's called the endomembrane system. So now, right underneath the cell membrane is the cytoskeleton, and it's made up of 3 different fibers. You have the first one, which is microtubule, and these are pictures to show you the microtubule. So if you look on here, you can see all the yellow lines, and that's essentially the microtubules. The intermediate filaments are the red things, and then on here, the microfilaments are going to be the lines. So you get these combination of these different structures that help with either creating like the scaffold to help support. It's like the building that you make whenever you look at the scaffolding of the building, and then we end up putting walls over it and everything else. It's the same idea with the cytoskeleton. It also helps things attach. So if you look in this cartoon here, you'll notice that underneath, you see on the outside, where the outside of the cell is, you'll see the plasma membrane, and then you'll see 3 different structures. So they are important for establishing the shape of the cell and providing strength and locomotion. So essentially, if you look here, these are microtubules. They are the largest, and they help with stability, but basically, you can have vesicles walk back and forth on these things, and I'm going to show you a video in class that demonstrates this, and it's pretty an amazing gig there. So basically, you have motoproteins that are like little feet that carry vesicles back and forth in the cell. So they get generated and tell basically where the vesicle needs to go, and they're little highways. If you see here, this is the intermediate filaments, and those are the second ones. So that's basically going to take the microtubules that they're there for support and attach them to proteins on the membrane itself. And then these really small guys there, those lines, those are the microfilaments. So you can think micro and filaments are normally really small. And so these help with separating essentially chromosomes when we do cell division, which is mitosis and meiosis, and they help for organelle transfer. So this just goes, the microtubules are the first ones I talked about. They are considered, they have dynamic instability, which means they form and then they're torn down. Intermediate filaments are rope-like, and actin filaments, which are also known as microfilaments, they are the longest, but they're very, very thin. And so that's just kind of showing you some more pictures. Now within those, within the membrane, there are also different types of kind of these things that help to attach. So desmosomes are these guys. You can see they are located right here. And they're going to help attach one cell to the next, right? The next kind of junction between cells is what's known as a gap junction. So on here, if you have any kind of ions on over here, say sodium, sodium can go through these holes. They're holes or channels to get from one side to the next. You see these a lot in nerve cells. And tight junctions are essentially these things here that make it so essentially you bind cells together. So these are different from essentially desmosomes because these guys are almost like waterproofing. Nothing can get through these. Where in desmosomes, they're considered like molecular Velcro. Let me take you back really quick so you can see them. Do you see how in those guys here, there are these things that kind of create these areas in the middle? So they can they give some flexibility. They give them so that if there's a lot of tension against them, they can relieve some of the tension. Where in tight junctions, you can see they're really close together and those ions and water and everything else can't get through. So these are going to be something that you would see in something like the digestive system where you don't want any kind of water or anything to leak out between the cells. Now the last part of this is the endomembrane system. And essentially this is a way that when we make proteins in the rough ER, that they can end up getting and being distributed into the correct cellular compartments. So it includes the nuclear envelope, the rough ER, the Golgi, and lysosomes and vesicles. So let's look at this really quick. So essentially what happens here is that you end up getting in the nucleus, the DNA essentially makes it to RNA, which we'll talk about that later, but you get the instructions to how to make a protein. And then essentially the instructions come out here, it attaches to one of the ribosomes, and then you can see these little dots. We make proteins. Well, essentially this is going to go through either keeping it the same or changing a little bit until it gets to the end, and then it's going to make a vesicle. The vesicle is going to move its way to the Golgi, come into the cis side, the same side as the nucleus, and then once it gets into the Golgi, it's going to move, and at this point the protein is made, but we might modify it a bit more here. Now from here, you essentially get this modified protein, and then one or two things can happen. If it's ready to use, it's going to go into a vesicle again, and then essentially it can be incorporated into some kind of organelle in the membrane, or it can be released out into the what's known as extracellular matrix. Think extra as in exit or outside the cell, right? If you make the protein and it's no good, then it can be put into a lysosome where you essentially eat it all up. So the best example I can give to you guys here is if just all of a sudden you're like, you know what, I want to make teddy bears. The instructions on how to make the teddy bears would be made inside the nucleus. And then you would move them over to the rough ER, where then they would make the teddy bears. Once they're there, they can make the teddy bears, but maybe they decide, okay, we don't just want, say, regular brown teddy bears, but maybe we want purple teddy bears or, white teddy bears or green teddy bears, whatever. They'll change those up a little bit, and then they'll put it into a truck. And then the truck will be sent to the Golgi here, right? And here they'll say, okay, we have the teddy bear, but maybe we want to make them add things to them to make them a football teddy bear. And so they put the little teddy bear in a Green Bay Packer jersey with a helmet, and then they put him again into these trucks, and then they send the truck to the destination. Now, if they take the teddy bear out and the teddy bear's a mess and it's all messed up, then you'll take it to the dump where it'll be put in a trash compactor and destroyed so that you can reuse the parts or destroy it forever. So that's the best example that I can give to you about how the endomembrane system works. And this is just showing you that essentially it goes from the rough ER into little vesicles into the Golgi, and then it can either be secreted, put into the plasma membrane, or digested and thrown away. Now that's all I have for the lecture. What I do want you to watch is about 3 minutes of this, about how biology and science can be beautiful. And at the end of this video, I'm going to tell you a little bit about what you expect to see in after this.
I'm a medical illustrator, and I come from a slightly different point of view. I've been watching since I grew up the expressions of truth and beauty in the arts and truth and beauty in the sciences. And while these are both wonderful things in their own right, they both have very wonderful things going for them. Truth and beauty as ideals that can be looked at by the sciences and by math are almost like the ideal conjoined twins that a scientist would want to date. These are expressions of truth as awful things, by meaning they are things you can worship. They are ideals that are powerful, they are irreducible, they are unique, they are useful, sometimes often a long time after the fact. And you can actually roll some of the pictures now, because I don't want to look at me on the screen. Truth and beauty are things that are often opaque to people who are not in the sciences. They are things that are that describe beauty in a way that is often only accessible if you understand the language and the syntax of the person who studies the subject in which truth and beauty is expressed. If you look at the math E equals mc squared, if you look at the cosmological constant where there's an anthropic ideal where you see that life had to evolve from the numbers that describe the universe, these are things that are really difficult to understand. And what I've tried to do since I had my training as a medical illustrator, since I was taught animation by my father, who was a sculptor and my visual mentor, I wanted to figure out a way to help people understand truth and beauty in the biological sciences by using animation, by using pictures, by telling stories, so that the things that are not necessarily evident to people can be brought forth and can be taught and can be understood. And students today are often immersed in an environment where what they learn is subjects that have truth and beauty embedded in them, but the way they're taught is compartmentalized, and it's drawn down to the point where the truth and beauty are not always evident. It's almost like that old recipe for chicken soup, where you boil the chicken until the flavor is just gone. And we don't want to do that to our students. So we have an opportunity to really open up education. And I had a telephone call from Robert Liu at Harvard in the molecular and cellular biology department a couple of years ago. And he asked me if my team and I would be interested and willing to really change how medical and scientific education is done at Harvard. And so we embarked on a project that would explore the cell, that it would explore the truth and beauty inherent in molecular and cellular biology, so that students could understand a larger picture that they could hang all of these facts on, that they could have a mental image of the cell as a large, bustling, hugely complicated city that's occupied by micro-machines. And these micro-machines really are at the heart of life. These micro-machines, which are the envy of nanotechnologists the world over, are self-directed, powerful, precise, accurate devices that are made out of strings of amino acids. And these micro-machines power how a cell moves. They power how a cell replicates. They power our hearts. They power our minds. And so what we wanted to do was we wanted to figure out how we could make this story into an animation that would be the centerpiece of BioVisions at Harvard, which is a website that Harvard has for its molecular and cellular biology students that will, in addition to all the textual information, in addition to all the didactic stuff, put everything together visually so that these students would have an internalized view of what a cell really is in all of its truth and beauty, and be able to study with this view in mind so that their imaginations would be sparked, so that their passions would be sparked, and so that they would be able to go on and use these visions in their head to make new discoveries and to be able to find out really how life works. So we set out by...
Okay, so I showed you that so that maybe you could get an idea that cells and biology are really beautiful. And some of the images that he shows on this talk are absolutely spectacular. And I actually have this video that they created, which to me, the first time I saw it, I was newly out of undergrad and I had just started working on my PhD, and it was absolutely breathtaking to me because the first time in my life I actually got to see visually what was happening in a cell, and it was just kind of mind-blowing to me because at that point when I was in school, most of what I'd seen were just cartoons, pictures, and a piece of paper that made it seem very dry and very much disconnected me from the process. So once we get done with everything that we need to do, I'm going to actually pull this out at the end of this subject matter lecture and show this video to you guys. And while you are watching it, I would hope that you would ask yourself, do I know what organelle that is? Do I know what function that is? And be able to quiz yourself as you go through to see if you can recognize some of what's happening. So I hope you guys have a wonderful night, and we essentially are finished with our introductory lecture on the cell.